Okay, so I did my term project on the Coronaviridae viral family. The Coronaviridae family consists of positive, single-stranded, large RNA viruses with envelopes, and their taxonomy places them in nidovirales. They are actually the largest known RNA viruses, with genomes ranging 25 to 32 kb, virons of 118 to 140 nm in diameter, and there are two subfamilies to the Coronaviridae. We have the Torovirinae, which has a helical nucleocapsid, and the Coronavirinae, which has a spherical nucleocapsid. I'll be focusing on the Coronavirinae subfamily, and within that subfamily, the SARS coronavirus 2, severe acute respiratory syndrome coronavirus 2, which is our current pandemic. So it, as we all know, enters through the nose, airway, and lungs until it meets an ACE2 receptor cell, and then it binds in that cell with the S spike protein, enters and makes many copies of itself. And we see a lot of those ACE2 receptor cells specifically in your heart, kidney, and gut, which is one of the reasons why it's causing chronic inflammation and can cause lots of pain in those areas. So the copies that are being made within the cell break out and infect other cells before the immune system is alerted, because SARS coronavirus 2 and especially the newer variants can prevent and interpret those signals to the immune system, which is really unique. And that's why you can infect other people before you even show symptoms. So the mutation rates of SARS coronavirus 2 make it most similar to AIDS and hepatitis C, like how fast the genome replicates is most similar to AIDS. All Coronaviridae viruses share the ability to move their polymerase complex during mRNA synthesis. This is another reason we see so many variants. That's very unique. Just to be clear, AIDS and hepatitis C don't do that, but they're also fast. So multiple highly divergent variants of SARS coronavirus 2 are being seen, like Omicron, Delta, Alpha. So a large number of amino acid changes and mutations within chronically affected hosts, so hosts that remain in the acute stage of infection for more than 5 or 6 weeks. Because it stays in the host so long, all these mutations happen. But the interesting thing is that the variants that are emerged from these acutely affected hosts are passed to other healthy hosts. So let's talk about my painting. Here it is. I tried to make it personal because this pandemic has affected my work and my life so much. So first of all, let's really focus on this S spike protein that's binding to those receptors, letting it sneak in and control 60% of affected cells' activity, which is insane. So Delta and Omicron variants have mutations in part of the S spike protein that bind to a cell, and these mutations help the virus completely evade antibodies. Terrible news. So RNA is single-stranded in red. I made it red so you can see it clearly. And then we have our N protein in blue, and that's creating this helix type structure here. The heart was just an artistic choice, but ignore that. This protein is responsible for identifying and wrapping the virus RNA into a helical structure. It also forms ribonucleoproteins, which are really cool. And then we have our E protein in yellow. So it's kind of embedded in the envelope, in the lipid envelope. And it self-oligomerizes to form a pentameric ion channel, making it a viroporin, and it triggers that inflammation in the host. And then we have our M protein in purple, also in the envelope. And it's an abundant protein. So the closest known relative of the airborne SARS coronavirus 2 strain causing the current global pandemic was found in a bat in China, as we know, and that strain was RATG13. But I wanted to talk about how the species currently displaying the most carriage of coronavirus besides humans is the Canadian white-tailed deer, which is actually present in parts of Oregon. Maybe a reason not to go hunting this year if you're into that, at least for white-tailed deer. And 1 in 3 people, so we've made a jump from 1 in 5, 1 in 3 people will be experiencing long COVID. And we don't really know what that means yet.